Hi kids! So we are now at the last stage of our carbohydrate catabolism or metabolism, and these are your electron transport chain and your oxidative phosphorylation. Now as you can see, we will also include in our discussion your pentose phosphate pathway. So going back to our discussion regarding glycolysis and citric acid cycle, so just to total all the ATPs and coenzymes that had been generated in these two pathways, that would be 4 ATPs, 10 NADH, and 2 FADH2. And with your coenzymes, NADH and FADH2, so these coenzymes will proceed to our next stage, and that is your ETC and your oxidative phosphorylation. So, the same with your citric acid cycle, your oxidative phosphorylation and ETC will still happen inside your mitochondria. So, the only pathway that is in the cytoplasm is your glycolysis, and that glycolysis is also the only anaerobic process in your carbohydrate catabolism. So, recall also here the requirements and the result in each of your stages and pathways. And for your stage 4, for our oxidative and ETC, so again, our primary requirement is whatever the products that had been produced in your citric acid cycle. And our result here would be the formation of your ATP, including the formation of your water molecule. So you'll get to understand that better as we go along our discussion. So let us define what is electron transport chain first. Now, from the name itself, kids, that is already the purpose or function of this carbohydrate metabolism, wherein there is simply a transport of your electrons from one complex or protein matrix to the other. And as defined, this is a process by which your hydrogen ions and electrons from your NADH and FADH2 are passed from one electron acceptor or carrier to the next, until such time that it will combine with your oxygen in order to form your water molecule. Now, regarding the position of your electron transport chain, it is here buried in your inner membranes, and comparing it to your citric acid cycle, which is this one, so it is basically in the matrix portion of your mitochondria. So ito po yung ating, this portion is our ETC and oxidative phosphorylation, while your citric acid cycle is on the matrix. So going back to this image, dito po na area, this is what we call your matrix. This is where your citric acid cycle will happen. So each of these complexes, each of these structures that you see that are buried in the cell membrane of our mitochondria, these are different complex proteins, and each of these complex proteins have a different name or another name, and these are their other names. So for your complex 1, we have your NADH coenzyme Q reductase. Complex 2 is your succinate coenzyme Q reductase. Complex 3 is your coenzyme Q cytochrome C reductase, and complex 4 is your cytochrome C oxidase. And this is what is exactly happening inside each of these complexes. But before we proceed discussing the steps that is happening in each of your complexes, take note that there are 2 electron carriers here in our ETC. So you have your coenzyme Q and you have your cytochrome C. Both of these will serve as a mobile electron carriers that shuttle electrons between various complexes. Now, the letter Q here in your coenzyme Q represents the presence of your quinone molecule. So it is actually just like your coenzyme A. Na letter A would represent its function na sa pag-transfer ng acetyl group nyo in order to proceed with our citric acid cycle. So again, hindi po siya meaning, representation lang yung mga letters. So let us discuss your complex 1. That is again your NADH coenzyme Q reductase. Now, all of the processes that is happening here in your different complexes, from complex 1 to complex 4, everything is in redox reaction. So unlike your glycolysis and your citric acid cycle, where there are a lot of processes that is happening from your phosphorylation to your isomerization and such, again here in your electron transport chain, it is only your oxidation reduction. Now, in remembering the names of your complexes, kids, the first name will always be your primary requirement, and the last is regarding the product or the ending substance that will carry your electrons. And looking at the different enzymatic steps here in your complex 1, this is referring to the electrons that is being transferred from one molecule to the next until such time that it will be received by your coenzyme Q. So, take note that the electron that is being carried here are 2 by 2s. So, dalawang electron, and in every electron that is being carried, hydrogen ions are being released into your intermembrane spaces. So, NADH, again, it comes from the citric acid cycle, which is our product. And what will happen here, sunod-sunod lang, kids, again, just observe how your electrons are being transferred from one to the next. So, NADH will first transfer electrons to your FMN. FMN stands for your flavine mononucleotide. Now, your flavine mononucleotide, somewhat mura lang ni sila og igsuon sa inyuhang FAD, your flavine adenine dinucleotide, in which they differ in the number of phosphate group that is attached to your adenosine. For your flavine adenine dinucleotide, di ba di because referring to the presence of 2 phosphate group, but with your flavine mononucleotide there is only 1. But still, they are the same. They still contain your riboflavin. And this is also how your coenzyme Q would look like in terms of its structure, and this is the specific portion in your coenzyme Q where your hydrogen ions will attach to. So going back here in our reaction, so again, the first electron acceptor after your DNH would be your FMN, and the next would be your FESP. Now, your FESP stands for your iron sulfur protein. And as you can see, there are numerical designations here in your FESP, and this is actually referring to your ferric and ferrous, where it is oxidized and reduced. So, Fe3 is your ferric, Fe2 is your ferrous. Ferric will be reduced to ferrous, ferrous will be oxidized to become your ferric. And then again, our last would be your coenzyme Q. So now let us proceed to your second complex. And as you can see, once we already have your coenzyme Q, here in your complex 1, it will directly proceed to your complex 3. So for your complex 2, we will need to create another coenzyme Q that will proceed with your complex 3. And what is happening here in your complex 2, we will begin with your FADH2. Now you might wonder, na ma'am, as you've said, ang first word po man kaya is relating to the first na product or molecule that will be used in the complex. And why is it that this complex is called succinate, where in fact what we are using is your FADH2? Now, again, FADH2 comes from your citric acid cycle. Now, here in the specific step in your citric acid cycle, there we will find the production of your FADH2. And as you can see here in the enzymatic reaction, our substrate is your succinate, and then reducing your FAD, and then transforming it to become your fumarate. So thus, in this complex, we call it succinate coenzyme Q, succinate referring to the substrate where we were able to get your FADH2. And here in your complex 2, your FADH2 can directly give or transport its electrons to your FESP until it is received by your coenzyme Q. Now, your NADH kasi kids is considered to have a high redox potential, which is not equivalent with your FESP, and thus it needs to transport the electrons first to FMN, and FMN will be responsible in transporting the electrons to your FESP. Unlike FADH2, which have the same redox potential with your FESP, thus it can directly transport its electrons. So just to summarize, your complex 1 and complex 2, again, they differ in the substrate that has been used. For complex 1, that is your NADH. For your complex 2, that is your FADH2. But either your complex 1 and complex 2 will end up with the coenzyme Q na electron carrier. And as you can see here in your complex 1, again, there is involvement with your flavine mononucleotide, but for your FAD, it is directly transferring electrons to your FASP. So, going back to this image, so again, complex 1, hindi po siya dadaan sa complex 2. Complex 1, by the time it will produce or by the time it will give its electron to your coenzyme Q, it will directly go here to your complex 3. And here in your complex 2, coenzyme Q pa rin ang ending going to your complex 3. Now remember that in each of these complexes, they are carrying 2 electrons. So by the time that we will form 2 coenzyme Qs here, one from your complex 1 and another from your complex 2, the starting of your complex 3 will carry 4 electrons. So here in our complex 3, we call it your coenzyme Q cytochrome C reductase. So as you can see, all you have to remember here is ang sunod-sunod na transferring sa electrons. So from your coenzyme Q, going to your FASP, going to your cytochrome B, going to another FASP, received by your cytochrome C1 until it will be carried by your cytochrome C. And among all the cytochromes that we have mentioned, di ba natay cytochrome B, natay cytochrome C1, your cytochrome C is the only cytochrome that is water soluble. And that is why in terms of how your cytochrome C is positioned, it is actually positioned in such a way that it is exposed to water. Because then again, it is water soluble, so meaning it is hydrophilic. And remember that in this portion, sa phospholipids nyo kids, the tail is hydrophilic, while the head is the one that is hydrophilic. Again, the tail is hydrophobic, and the head is the one that is hydrophilic. So thus it is positioned in such a way that it is exposed to water. Now, another reason why it is placed in that area, it is stated that parang factor daw siya. It is a factor for the mobility of your cytochrome C so that your cytochrome C could move freely within your intermembrane space and pass the electrons that it got from complex 3 to complex 4. So here in our complex 4, this is what we call your cytochrome C oxidase. Just remember the word oxidase because this time around you'll get to see the presence of your oxygen. So what happens here, kids? Again, just remember how electrons are transported. Now here in your cytochrome C oxidase, it contains 2 different heme centers. Now remember that cytochromes are examples of substances with a heme content in its structure, and the 2 heme centers that we are pertaining here is your cytochrome A and your cytochrome A3. Other than having the iron inside the heme, it also contains your copper na molecule. So that is why here, may copper A, may copper B. So, in terms of transferring electrons from your cytochrome C to your cytochrome A, particularly, dadaan po muna kay copper A and then kay iron sa inner portion ng cytochrome A nyo. But here in your cytochrome A3, it will transport directly its electrons to your copper B. And then again, what we are carrying here are a total of 4 electrons because 4 electrons are needed to reduce your oxygen to become your water molecule. So oxygen will then be reduced and will become your water. And what happens here, by the time na natay water, kids, it will just be released into the circulation. So the water molecule here, consider this as a waste product that needs to be excreted. So thus, oxygen rather. So thus, take note, oxygen is considered to be your final electron acceptor here in our electron transport chain. So again, it is estimated that 95% of your oxygen is used by cells to serve as our final electron acceptor. And that is why it is really important to have a good amount of oxygen, and that oxygen is being delivered by your hemoglobin, because without your oxygen, ATPs cannot be produced, your electron transport chain, and even your citric acid cycle will never be able to proceed. So again, if there is oxygen deprivation, then another cycle will be activated, and that is your Cori cycle in relation to the conversion of your pyruvate to become lactate. So again, yan yung last na ng inyong electron transport chain. So this is the summary of your electron transport chain. And again, balik-balikon yun ako, kids. All you have to really remember is in terms of the transport of your electrons. Now, where does oxidative phosphorylation happen? Oxidative phosphorylations happen in a different na complex, and this is our complex 5. And complex 5 is considered to be your ATP synthase. But before we further discuss about the action or what is happening here in your oxidative phosphorylation, let us define what is oxidative phosphorylation first. So oxidative phosphorylation is a series of reactions which couples the oxidation of your NADH and FADH2 to the phosphorylation of your ADP to generate now ATP. So kumbaga, that is where your ATPs will really be formed rather than in your ETC. So kaya kids, kung makikita nyo, while we were discussing about what exactly is happening in your ETC, diba? Never natin na-mention na may formation ni ATP or even your GTP pa ba? Because your electron transport chain, naalam po na siya different na function or importance. But still, even if there is no ATPs generated here, again, nani siya dako na role para mag-start inyuhang oxidative phosphorylation. So, ipadayon sa nako, kids. Now, here in your oxidative phosphorylation, other than the formation of your ATP, we will be able to convert your coenzymes to ATPs. And in every NADH that is formed, we can produce around 3 ATPs. And for your FADH2, we can produce around 2 ATPs. So, this is how important electron transport chain is. Looking at this image, kids, and as what I have mentioned a while ago, the importance of your electron transport chain is in terms of the release of your hydrogen ions here in the intermembrane space. It is actually very important because without a sudden increase in the hydrogen ions present in your intermembrane space, your oxidative phosphorylation will never begin. Because here in our mitochondria, we always need, or not just your mitochondria, but diba, in our body kasi kids, we need to maintain this what we call equilibrium. So meaning at any compartment in our body or in our cell, there should be equal amount of substances outside and inside. So what will trigger the phosphorylation of your ADP to become your ATP is the difference in the concentration gradient here in your intermembrane spaces and here in your matrix. And as you can see here in this GIF, here in this GIF, this presents that in order for our intermembrane space and matrix to equalize, para ma-reach natin yung equilibrium, your hydrogen ions will move going to your complex 5, which is our ATP synthase, and as it moves down to your complex 5, a potential energy will be generated converting ADP to become your ATP. So, sadyang ingana lang yun siya, kids, na kumbaga, pagbaba lang yun sa inyong hydrogen ions, there will be a potential energy created by the electrochemical gradient by its movement from a higher concentration to a lower concentration, and that energy would trigger the phosphorylation of your ADP to ATP in the presence of your enzyme, your ATP synthase. Now remember, hydrogen ions, not just your hydrogen ions, but any molecules will really move to a certain area to equalize these two different areas or concentration, and that is what we call the diffusion. But here in the movement of your hydrogen ions entering your complex 5 in order to generate the energy, we call it your chemiosmosis. So I hope na gets siya na again, kailangan papasok yung hydrogen ions nyo to your complex 5 going to your matrix, that is to maintain equilibrium. And thus, important ang role ni ATC kasi siya yung nag-release every time we transport electrons from one complex to the other. Hydrogen ions are being released to your intermembrane space. So, nagtataas po talaga yung concentration ng hydrogen ions sa intermembrane spaces nyo. And because of the difference in the concentration gradient, your hydrogen ions will pass through your complex 5 to go to your matrix, and upon passing through your complex 5, energy will be created. Now, as you can see here in this image, may numbers po tayo na nakikita, and that is your F0 and your F1. So kindly take note that your ATP synthase, your complex 5, has two subunits, F0 and F1. F0 is part of your synthase, and it functions for the channeling of your hydrogen ions, whereas the formation of your ATP would take place in your F1. So yun yung kanilang purpose. Again, F0, where a channel for hydrogen flow. F1, formation of your ATPs. So kaya kids, going back to this image, so it is really pointed out here, yung chemiosmosis na referring sa movement na necessary na kailangan may movement talaga ng hydrogen ions nyo in order to start your oxidative phosphorylation and then creating now your ATPs. So to summarize everything here in your ETC, so let me just present this video first. So kindly watch this video, and then after which we will continue with our discussion.
All of the steps of aerobic respiration that we have done so far have been leading up to this, the electron transport chain. This is the main event, the big bonanza, the Super Bowl of cellular respiration. Why? Because the vast majority of our ATP is made here. The electron transport chain is actually a series of proteins and other molecules embedded in the inner membrane of the mitochondrion. These molecules transport electrons, hence the name. Let's zoom in on this inner membrane to see the electron transport chain in more detail. Here's how it works. NADH, that electron carrier we've been making all along, delivers its electrons to the first protein in the electron transport chain. As the electrons move through this protein complex, energy is gradually released, and this energy is used to pump hydrogen ions, or protons, across the inner membrane, from the matrix into the intermembrane space. At this point, it might help to realize that moving electrons is electricity. So it should not be surprising that these electrons can provide the energy to drive these microscopic pumps made out of protein. They are basically natural electric pumps, created way before humans invented them, which is super cool. The electrons are then transported by this special red molecule toward their next destination. The red molecule also picks up the electrons donated by FADH2, that other electron carrier that we made during the Krebs cycle. All of these electrons are then passed on to another protein complex. At this next protein complex, the electron energy is used once again to transport hydrogen ions across the membrane into the intermembrane space. Notice that we are building up a high concentration of hydrogen ions in the intermembrane space. This will be very important soon. Now the electrons are transported to a final protein complex, where their energy is used once again to pump hydrogen ions into the intermembrane space, making the concentration even higher. Now the electrons need somewhere to go, a final destination to complete their journey. Fortunately, you breathe, and oxygen comes to the rescue. Oxygen accepts the electrons, as well as hydrogens from the matrix, making water. This is the reason why you breathe all day, every day. Your cells need oxygen to accept these electrons, or the electron transport chain will grind to a halt, like an LA freeway during rush hour. And if the electron transport chain stops, you won't be able to make enough ATP to keep you alive, and you die. So keep breathing that oxygen. Now you may have noticed that we haven't made ATP yet, so you might be wondering how that works, which brings up the coolest part of this entire process in my opinion. Remember that high concentration of hydrogen ions we've been building up? Now it's time to use it to make ATP. A protein called ATP synthase makes a channel that allows hydrogen ions to flow through from high concentration to low concentration. You may remember that molecules tend to move from areas of high concentration to low concentration naturally, a process known as diffusion. So, these hydrogen ions, in a manner of speaking, want to flow from the high concentration area of the intermembrane space to the low concentration area, the matrix. But they can only get through ATP synthase. This facilitated diffusion of hydrogen ions through ATP synthase causes ATP synthase to spin like a turbine and produce ATP. Such a cool mechanism. It's like a microscopic hydroelectric dam, generating ATP instead of electricity as hydrogen ions passively flow through. Since we made many electron carriers throughout respiration, the electron transport chain happens over and over again, making between 30 to 34 ATP molecules for every glucose that entered respiration. If you are doing the respiration activity at Bioman Biology, you should now return to
Ayun kids, so that video summarizes everything that we have mentioned. And then again, I hope you get to appreciate na the very purpose of your ETC is really to release hydrogen ions. So we will intentionally increase the concentration of hydrogen ions in your intermembrane space because that is the only time that your ATP synthase, your complex 5, will be activated and will be able to generate ATPs. And then in terms of your oxygen, it is your final electron acceptor, and in order for it to be released into the circulation or excreted in your mitochondria, due to the presence of your 4 electrons, it will be reduced to become your water and then will be released out. Then again, in terms of your 4 electrons, remember that in your complex 1 and in your complex 2, each of those complexes transferred 2 electrons to your coenzyme Q, so therefore, your coenzyme Q will be carrying a total of 4 electrons. So going back here, in our carbohydrate metabolism, now again, let us now look at the number of ATPs and coenzymes that has been generated or produced. And to summarize everything, kids, in terms of how many ATPs are yielded at the end of your carbohydrate metabolism. So, ganito po siya. So, again, remember our ruling, every NADH times 3, every FADH2 times 2. But somehow, may kailangan po tayong tandaan dito. There is a difference in the number of ATPs produced if the NADH was created in either the cytoplasm or in the mitochondria. If it is a cytoplasmic NADH, times 2 lang po yung ating ATP na ma-produce. If the NADH is produced in the mitochondria, then we will multiply it by 3. Now, the only reason that was provided as to the difference of the number of ATPs in your NADH, sadyang ang na-check ko lang talaga, kids, is that dun kasi sa cytoplasm nyo, there is lesser energy production. So, kaya lesser din yung number of ATPs na ma-produced from your cytoplasmic NADH. So, kaya, if we will total everything, converting your NADH now to the ATPs, again, in your glycolysis, you already have 2 ATPs, and the 2 NADH there will be multiplied by 2, so we already have your 4. Now, in the conversion of your pyruvate to your acetyl coenzyme A, we also have 2 NADH. So this time, remember that the conversion of your pyruvate to acetyl coenzyme already happens inside your mitochondria. Pyruvate will enter into your mitochondria and then will be converted to acetyl coenzyme A to participate in your citric acid cycle. So that is why the NADH here, tatlo din po yung ma-produce. So we have 6. And then for your citric acid cycle, we have 3 products, and your first product is your GTP, which is just equivalent to your ATP. Another form of energy lang din po siya. So as is, that is 2. your 6 NADH multiplied by 3, so that is 18, and then your 2 FADH2 multiplied by 2, then that is 4. So if we try to total everything, kids, in relation to this image, then our total number of ATPs that we will get from your overall carbohydrate metabolism is 36. So, comparing it now to what is happening when there is oxygen deprivation. So, ito yung sinasabi ko na hindi pwede na hindi makorek yung oxygen deprivation ng isang individual because since it will not begin your citric acid cycle being an aerobic process, yung number of ATPs din na mag-give off ng pyruvate to lactate conversion nyo, yung Cori cycle, is only around 2 ATPs. So, imagine the difference in energy kung magsigilag Cori cycle comparing it to your overall carbohydrate metabolism na 36. So, we are done with your carbohydrate metabolism. So, some additional pathways na lang din, kids. Now, this one, we will call this your malate-aspartate shuttle because what is happening here is the interconversion of your malate to aspartate and vice versa with the involvement of your oxaloacetate. Now, malate-aspartate shuttle is very important especially in tissues with high energy demand. So, ano yung mga tissues na may high energy demand? That would be your heart, your kidneys, and in your liver. Now, the very purpose of this pathway or shuttle is to give additional energy for those tissues because yung mga liver, heart, and kidneys natin among all of the tissues or organs in our body, diba sila man gid ang naay pinaka-grabe na trabaho. And so to give additional energy from the 36, your malate-aspartate shuttle will also be involved for the production of ATP. So, what will happen here, kids? Looking at your oxaloacetate, remember that this is the end product of your citric acid cycle, which will then be reused in order to continue the cycle. Now, oxaloacetate will be used to convert malate because NADH kasi daw, kids, cannot directly enter into your mitochondria. So, for your NADH to be used here in your mitochondria, oxaloacetate should be converted first to malate. Malate will enter into your mitochondria, and then malate will then just be converted back to oxaloacetate to produce now your NADH. And since NADH produced here is only 1, then there is additional 3 ATPs in your malate-aspartate shuttle. Now, once we already have your NADH, we already have your ATP, oxaloacetate can then just be recycled. So, in order for it to go back to your cytoplasm, it will be converted to aspartate. Aspartate will go out to your mitochondria and will be converted to oxaloacetate, then participate po ulit sa citric acid cycle nyo. So, please be mindful of the enzymes that catalyzes each of this reaction. You have your malate dehydrogenase and you have your aspartate transaminase. So, last portion of our discussion would be your pentose phosphate pathway. So, what exactly is your pentose phosphate pathway? What is its role? So, why do we need this pathway? So, the other name of your pentose phosphate pathway is hexose monophosphate shunt. And this is actually an alternative pathway lang po for your glucose oxidation. And these are its purpose. Number 1, in terms of the creation of your NADPH. And the second is for your ribose 5-phosphate. NADPH, kids, just add the letter P dun sa definition ng NAD natin, and this represents your phosphate. Now, I pointed this one out in our discussion sa glycolysis na dun sa portion ng mga intermediate compounds nyo na sometimes an additional phosphate is placed in your ribose sugar and we call it now your NADP. So that is nicotinamide adenine dinucleotide phosphate. If there is presence of your hydrogen referring to its reduced form, so just like your NADH and your FADH2, so just add the word reduced. So that is reduced nicotinamide adenine dinucleotide phosphate. Now, the purpose of synthesizing NADPH is that it is an important coenzyme for the synthesis of your lipids. Whereas for your ribose 5-phosphate, it is important, especially in the synthesis of your nucleic acid and many other coenzymes. So looking at our enzymatic reaction here, our pathway, your pentose phosphate pathway will still make use of your glucose. And by the time it will be converted to glucose 6-phosphate, so ibang destination kaagad si glucose 6-phosphate. So these are the step-by-step enzymatic reactions that is happening in your pentose phosphate pathway. Take note that your pentose phosphate pathway is composed of an oxidative phase and a non-oxidative phase. Now, the oxidative phase is referring to the synthesis of our NADPH and ribose 5-phosphate. Pag non-oxidative siya, kids, it will begin with your ribose 5-phosphate producing your fructose 6-phosphate and glyceraldehyde and will then participate in your glycolysis. So, yun yung siyang pentose phosphate pathway nyo, kids. Depende kung ano yung kailangan. Dalawa yung phases, oxidative and non-oxidative. So, let us begin with your oxidative phase. Now, here, your glucose 6-phosphate will be converted to your 6-phosphogluconolactone. And our enzyme here is your Z6PD, your glucose 6-phosphate dehydrogenase. So, here, may first na tayo na NADPH production. Your 6-phosphogluconolactone will then converted to 6-phosphogluconate, and our enzyme is your lactonase. And then, your 6-phosphogluconate will be converted to your ribulose 5-phosphate, generating another NADPH. Now, your ribulose 5-phosphate will be converted to ribose 5 by your enzyme phosphopentose isomerase, and then ribose 5-phosphate will then be used for nucleotide synthesis. As to your non-oxidative phase, kids, again, it will begin with your ribose 5-phosphate, and then these are the step-by-step enzymatic reactions until now we will get these two substances that participates in your glycolysis. So basically, the purpose of your non-oxidative phase is to create additional glyceraldehyde 3-phosphate and fructose 6-phosphate to participate in your glycolysis. And there are actually two enzymes involved lang here. You have your trans-aldolase and you have your trans-ketolase. But para hindi na complicated, kids, ito na lang yung tatandaan nyo. When we are proceeding with non-oxidative phase, these are the intermediate compounds, and this is the enzyme needed, and then our product are these two. So, yun na lang tatandaan nyo. Si trans-ketolase lang na enzyme, and then may formation nila erythrose, cellulose, and your pseudoheptulose until such time na makuha po natin itong dalawa na needed also for your glycolysis. So, this table is just to present all the necessary substrates and products and enzymes in your oxidative phase ng kids sa oxidative phase ng ito na portion, oxidative phase in your pentose phosphate pathway. So that ends our discussion regarding your ETC, oxidative phosphorylation, and pentose phosphate. So if you have any questions, then do not hesitate to contact me. I hope you learned a lot and thank you for listening, kids. Bye-bye.